Hey, thanks everybody for being here today. It's my pleasure to introduce the MindCORE seminar speaker, Kyle Berger. Um, Kyle has many letters after his name, and some of those letters I had to look up what they meant. So he's not only has a PhD, he also has a master's in public health, and the RD is he's a registered dietitian. Did I get that right? Okay, yeah. And so you could imagine, given that mix of letters, that he's very interested in things like uh, eating behavior, ingestive behavior, and what he has, what a lot of his research has involved is neuroimaging of food reward, neuroimaging of cues for food reward. And as you can imagine, there's a lot of very interesting questions uh, involved in that are around those issues, things like, is food addictive? Should we be worried about advertising, and does that contribute to the obesity epidemic, etc. Part of the reason for inviting Kyle to speak here to MindCORE to speak to us today is that he is in the middle of a move from his current position as a professor at the University of North Carolina to the Monell Chemical Senses Center. Some of you may know about Monell, but it is right. I mean, next door is maybe even too far, right? It's basically on campus. If you've seen the sculpture of the human nose and mouth that exists at about 35th and Market, that is the marker that's right in front of the Monell Chemical Senses Center. And so Kyle is going to be leading up a neuroimaging effort at Monell once he gets here, and we're really excited about that. And we wanted to introduce him to the community and hear about his work. And I'm going to hand it over to Kyle.
Thanks so much. All righty. Well, thanks everyone for coming. I always enjoy doing lunchtime seminars so people feel really awkward as they're eating. Yeah, no. So let's just kind of jump right in. Just to kind of give you the roadmap. First, I'm going to talk about just some of the basic imaging methods that we do. Some of you might be already familiar with that. And then I'll talk a little bit about theories of food reward and body weight. It's been studied for about 20 years now, 30 years. So not that long in the grand scheme of things if you compare it to substance use disorders or those kind of things. Then we'll focus on more of a food reinforcement or ingestive behavior, and then just kind of quickly wrap up. First, some of our imaging methods. This is a newer application for us. We've recently started doing neuromelanin sensitive MRI. And so neuromelanin is just a dark pigment. It's a byproduct of dopamine metabolism. It's non-invasive. It's essentially a structural scan. It's about 12 minutes. And it's a measure of, it's non-invasive in the SN and the VTA, so the dopaminergic midbrain. And this has been validated and primarily used in Parkinson's research, where excess neuromelanin is associated with Parkinson's, or and some in cocaine research, but that's really recent, like last year or two. So our functional imaging paradigms, really straightforward fMRI paradigms. For a long time, people would do food pictures. It's really easy. You're just showing them images in the scan. And, you always have a contrast image. So we have, highly energy dense foods or really palatable foods that people like, and compare that to fruits or vegetables, or, scrambled images, just so you get the bold contrast of like high palatability or high energy density. The other one that we do is a passive cue taste paradigm. So what you'll see is kind of a timeline. A would be, we show a symbol, which actually I think that's a symbol from Lord of the Rings because we're nerds. And then you have a little bit of break and we administer taste, whether that be milkshake, quinine, Kool-Aid, whatever we want to do, however we're trying to reinforce them. A little break, we have a swallow cue, so we know when to pop out head motion for that, and then they get a little rinse. And then you have our control, which is similar, different symbol. We use primarily a tasteless solution, which matches the osmolality of saliva. We clearly don't tell people that. We just say it's a tasteless solution. And then there's our contrast. And from this, you can get cue elicited anticipation of the beverage, and then also response to the beverage. And this is what it looks like in a first person experience. And that's like Sanjay Gupta in my forearms. I know, right? I would recommend not putting the GoPro in the magnet. But it wasn't my decision. They had a mock scanner too. I have no idea what it did, but hey, not my thing. So yeah, so that's just kind of very basic imaging protocols I just kind of wanted to walk through quickly. All right, so let's talk about some theories of food reward and body weight. And the one thing, so theories, the one thing I know about theories is theories are like toothbrushes. Everybody has their own, no one wants anything to do with anyone else's. So I'll just go over maybe the top 6 or 7 for the food reward brain-based theories of obesity. They fall in 2 categories, primarily kind of this heightened response to cues that's driving, wanting or acquisition and intake, or a reduced dopaminergic functioning. And those are primarily PET studies. And the cue studies tend to be functional. So first one is this, people are just born with this high response to food cues, like visual cortex, visual processing, ACC, so kind of attention, striatal response, these kind of things. But that's been going on and everyone just kind of assumed that's the way it is. But there's a lot of meta-analyses lately, finally, that we have like meaningful sample sizes that might be suggesting otherwise, or it might be a little bit more complex, identifying individual differences and such. Lastly, oh no, not lastly, this one is, again, you're born with this inherent striatal response to taste, and that actually predicts future weight gain. And as you're gaining weight, your response to the cues increases over time, and a little bit that your response to taste stays flat or declines over time with the excess body weight. And the nice thing about this is we did like a high-low risk model as on both of these studies, and we replicated the findings, different magnet, different part of the country, different sample. So we feel like that effect's relatively stable. What the youth high risk for obesity is actually maternal obesity when the kids are within like a recommended range of BMI. So that is actually the most robust, like maternal obesity is actually the most robust predictor of future weight gain across the board. Last one, you might recognize this if you do substance abuse work, is that food cues are sensitized through consumption, and that creates, wanting, not necessarily liking. And you'll see a theme of these, that they're basically almost find and replace from substance use theories, right? Obviously Kent Barrett has been long theorizing for substance use, and also kind of talked about some obesity and eating disorders. And I don't know about that other one that's talking about food abuse, but that was one of mine from recently. Okay, so on the reduced dopaminergic functioning on that side, like I said, these are primarily PET. So similar to that kind of innate response to food cues, this is, you just have compromised basal dopamine functioning. And therefore that equals kind of, you're trying to achieve some level of reward, and that could include a food reward seeking. So that uncomfortableness or lack of dopaminergic functioning and hedonically motivated behavior is actually pushing you towards pushing us towards a reward. This is similar. This is from Nora Volkow and Jean-Jacques Wang, that again, there's just like the low basal dopaminergic functioning leading to an impulsivity, because, striatal, projects to prefrontal cortex. And then there's also a high somatosensory response on oral somatosensation area. Now, this one's a little bit similar. This is by George Koob, these kind of things. And it is a little more dynamic in that a high food reward actually breaks the allostasis of dopaminergic circuitry, causing low basal dopamine functioning and capacity, which then results in additional food seeking. So those are like 6 of the 14 I can find. They're all within kind of this genre. And these are pretty common, and you can see there's a lot of overlap. So we're probably right somewhere in there. At least we're not wrong. Um, so the last one is this hyper and hypo, can we have it all? It's been recently hypothesized, even within PET studies, as a nonlinear relationship between basal dopaminergic functioning and body mass. So lower BMIs, um, increasing amount as your weight increases, increasing amount of dopaminergic functioning, and then it kind of falls off, which falls in line with kind of that George Cube dark side hypothesis of the system breaks and then goes back down. Um, and then the other thing that has suggested some of the inconsistencies of the findings is within the PET studies, there's the use of different ligands and different sampling frames. Some of the sampling frames, you can see like BMIs around 50, some of them BMIs around 30. Those are very different physiological states for sure. So we set out to do that neuromelanin sensitive MRI and see if we could detect this nonlinear relationship. Again, this like a byproduct reflective of dopamine metabolism, kind of aggregate dopamine metabolism. So just our regions of interest, we have the substantia nigra pars compacta, the pars reticulata, and then the ventral tegmental area. We include some others like the PBP and that kind of thing, but generally this region. And so what we found was just using a voxelized approach across that whole ROI, we found a significant effect, and it's both a positive and a nonlinear relationship. And you'll see we do permutations to determine significance. These are all significant voxels. You can see in the green, it's a lot of it is in the SNC. If we pop back through really quickly, and we had a decent sample size of about 58 or so. There we go. Yeah, if you just kind of keep an eye on the green, it's really predominantly in the green. So we wanted to do just kind of an ROI analysis of those regions independently. And we were right in that the SNC had the strongest effect. And you can see, it's if you're looking at effect size, it's not super strong. Behaviorally, that's not bad if you do the R on it, but it does detect a nonlinear and a positive relationship. So we know there's a strong relationship between high BMI and high impulsivity. So we had this data, so we wanted to see if there's neuromelanin moderated the relationship between BMI and cognitive performance using the stop signal task. We'll talk about the stop signal task a little more later on, but the two primary measures that we have for this are stop signal reaction time, which is response inhibition time, and then response inhibition accuracy. And we'll talk about that when we see the data. So for the VTA, there was not a statistically significant relationship, but it was P of 0.7. So there's probably something there. We're probably underpowered. And just kind of looking at the general, the way the effects are landing, you see that a lower BMI, neuromelanin was positively related to the SSRT. So in an area, there's more in the VTA, there's more dopamine metabolism that's associated with better response inhibition. So that's lower impulsivity. So, and then you can see in the higher BMI, it's inversely related to, so worse inhibition. When you're looking at response accuracy, that was a little bit stronger of effect. We saw it in the VTA as well as the SNC. Again, where lower BMI had, this is a different metric, so lower as less impulsivity, higher is less impulsivity. So the more neuromelanin in the SN and the VTA showed with better performance on the task. And then there's like absolutely no relationship between the higher BMI, and that's just really, really flat. So just kind of this piece in summary, neuromelanin reflective of dopamine metabolism showed associations with BMI that parallel PET measures of dopaminergic tone, particularly in the SNC. And there appears to be at least worth investigating an interaction of aspects of cognitive functioning and dopamine and BMI. And why this might be important is high BMI during middle age is associated with early cognitive decline. Once you get more elderly, it's higher BMI tends to be protective against a number of things, but during middle age, it's early onset cognitive decline. So thinking back just briefly to the food-related theories of obesity, they're all about obesity, and not the underlying behavior, which I always thought that was fascinating because all of them are all based off substance use disorder literature, which has nothing to do with obesity or altered neuroendocrine functioning associated with excess adipose tissue. They're all behavior focused. So why don't we start working backwards? And this is kind of how I started working backwards. Ingestive behavior, back to habit formation, back to decision making, back to reinforcement learning. It's kind of a process that, builds up and builds down. So now we're going to talk a little bit about food reinforcement and ingestive behavior. All right. Does this mean anything to anyone other than I would like to be in palm trees where it's warm? Yeah, so there's no context behind this. But if you're in Ireland, that is the second biggest fast food chain. And so I was fortunate enough to go, and we're running around, and there's billboards of just the palm tree. And that meant nothing to me because I had no idea what it was. There's no context. So the first thing that I wanted to look at briefly is one, whether we could replicate those response to cues and tastes associated with behavior and not necessarily excess adipose tissue. So we pulled 3 or 4 studies, all really cool, just kind of secondary data analysis, except for one. And basically summarize this, we saw objective measures of energy intake. So, using a doubly labeled water method, we saw that the more that these kids that were lean were eating, they showed greater response to the food cues. So just like you'd see in obesity, and in obesity, frequently you see a decreased striatal response to the taste. We did that with frequent ice cream consumption, which that one grabbed a little press, and it was hilarious because it'd be like, ice cream is addictive as crack. That was one of the headlines. Sure. Then we started looking a little bit about soft drink response, and we'll get into that a little bit here in a second. And this is the only public health slide I think I have for you, but I was thinking about a real world influence of ingestive behavior. So sugar is really cool, actually, because unlike fat or protein, you're born with an innate preference for sweet. And this is evolutionarily an advantage because what it's doing is it's signaling available calories. We can talk about artificial sweeteners at the end, but typically it's signaling calories. And if you look at sugar response in the brain, you'll see that it elicits, similar neural activity or, brain response in humans associated with hedonically motivated behaviors. And it's a nice primary reinforcer. Like a lot of people, use money as a reward and a magnet. But that's a secondary reinforcer. They can spend money on food. They could spend money gambling. So it's just one step removed. And when we're putting people in the magnet and they're tasting it, it's hard not to think about you're tasting something versus, your mind wandering about butter. So one of the things that we just kind of focused on, because it's a really easy public health target, is sugar sweetened beverages. And there are a ton of health risks. I mean, these are meta-analyses and dose response relationships between mortality, cancer, stroke, obesity, type 2 diabetes, et cetera. And despite all these known things, adults, 15 to 20% of their calories per day is coming from a sugar sweetened beverage. And on average, people consuming more than one. Obviously, that's average. We're thinking about population level studies with thousands of people. So, obviously you're going to have people driving the effect one way or another. But, come to think of despite all these known health risks, most people know that sugar sweetened beverages are probably not the best thing to drink. Why are people drinking them still? So probably has a lot to do with food branding and advertising. So last year, $1.5 billion in advertisements on sugar sweetened beverages alone. So in the US. So, and $14 billion on unhealthy foods total. So it's quite a bit of money, and they're very effective. Advertising research has been going on since the 30s. This is a great old book, The Hidden Persuaders, if you're interested in. One of my favorite quotes was, they're focused on manipulating human motivations to develop an internal need for a good, even if the person's disinterested in purchasing the product. And that was 1932 with that quote. My other favorite one was, the key strategy was to create a conditioned reflex where an individual responds automatically to flashing symbols or words without realizing it. So we'll see how that lands. Just looking at some fMRI literature as we're moving forward, responding to brands as kind of like this behavioral cue or advertisements. And viewing images of a luxury brand or your favorite product or car, those kind of things, increases bold response in striatum, visual cortex, precuneus, hippocampus. So, we're talking hedonically motivated behavior, attention, valence, memory, these kind of things. And you see a similar effect when you're showing like brands or logos in the magnet, particularly to children, which is a little more scary. One of the things that the first step that we started to do moving in the direction that I'm moving in was looking at the effect of consumption on this. So in this study, we had kids come in and they were either habitually drinking Coca-Cola, so more than 7 per week, or just like no sugar sweetened beverage at all, right? And this is, it's a very like dichotomous kind of pilot study, see if we get an effect. And in our picture paradigm, we showed them pictures of just the Coke brand without presenting any information on the product. But then we also showed them pictures of like advertisements that highlight the product. So like Coke, you can think of like the polar bears or something like that, or even just like the white wave. And there was no difference between those that consumed and those that not, if the product was shown. But if the brand was shown, there was a difference where those that consumed showed higher attentional and striatal response. So there's something to consumption. So then we start kind of asking, what's the mechanism? And we landed on maybe it's reinforcement learning, some of previous literature. The foremost of that is work by Wolfram Schultz and colleagues from the 80s through, I think, even published recently. But what this is, it's just a Bayesian conditioning experiment in a primate model, and those are dopaminergic recordings. And so what you see is when it's unconditioned, they have a cue, which is a light and / button. If you're not familiar with the work, there's no response to the cue, which is the orange, and then there's a dopaminergic response to the taste. And then after conditioning, you see that it flips, actually. So, and this is a lot of where prediction error kind of conversation started coming around. But you see dopaminergic response to the cue and now none to the taste. What's really cool about a handful of these studies is they correlated those dopaminergic responses with motivation toward the cue, whether it be like reaction time toward the button or willingness to work to receive the reward, those kind of things. And I think, both of these use juice in kind of a sugar sweetened beverage, which is friendly for the kind of work that we do. So the first thing we asked was this, I call it the Schultz shift of dopaminergic response, is that evident in humans? And obviously we're not measuring actual activity in fMRI, but you infer activity. We're not directly addressing dopaminergic response, although we're coming up with ways to maybe even do that. But we asked that question just in an fMRI experiment. Again, this is this kind of secondary data analysis. It's really fun when you're just kind of like walking around. You're like, oh, I might be able to do that with some old data. But you see that, especially in the VP and dorsal striatal response, the cue is unconditioned at early after 16 trials. It flips. In case you kind of wanted to look at it, these are just kind of parameter estimates from the cue on top, whereas early you can see there's less, a less bold response, and then later you can see a much greater bold response, and then for the taste you can kind of see that deteriorate. Those are unthresholded parameter estimates. If you're interested in how we did that, it's really actually quite easy. And what was nice about this sample was we actually followed them up for 2 years. and recorded their body weight and height for for those 2 years, like every 6 months or something like that. And what we found was that the stronger adaptive encoding of the cue predicted future weight gain. So those that are more readily sensitizing the food cue are more likely to gain weight over a 2-year follow-up. Which, if you can do prospective work in humans, that's always helpful. Then we asked, can we experimentally introduce this kind of food brand sensitization in a real world environment? So we did a randomized control trial with sugar sweetened beverages. And so here, just kind of worked through it, just to give you an idea of the measures. Oh yeah, we have a bunch of pre-registrations. We're happy to give code or data, just let me know. So what we did was, and I'll go through these, we did explicit visual analog scales for how much they liked it after they tasted it, just like 0 to 100. And of the sugar sweetened beverage and the unsweetened beverage, that's USB. We did some cognitive stop signal task, cognitive behavioral task, and then we did a brand taste fMRI study. Then we just randomized to sugar sweetened beverage group or unsweetened beverage group. They had 3 weeks of consumption that they consumed every day. So they came into our lab, they drank one in front of us, we sent them home with another one. They brought the bottle back for adherence, which isn't the best measure, but at minimum, we knew they were drinking it 3 times a week because we saw it. And then we did the same battery of assessments at the end. And then we also included a reversal learning fMRI task. So looking just at the beverages and the branding, those are super awesome brands. No, they're just meant to be as basic as we could. And those are stickers on the symbols. I mean, stickers on the beverages. Yeah, so the active sugar sweetened beverage condition, same amount of calories and sugars as a soft drink. They drank 10 ounces of it. Unsweetened control, no calories, no artificial sweeteners, and they were flavored the same. We did about a 2-month-long pilot study to determine the flavors that people would rate, as acceptable, that they're willing to drink every day for 3 weeks. Obviously, the participants can't be fully blinded in this situation because everyone can taste something that has sugar and something that doesn't. But we were blinded, so we could, well, at least analytics and up to me were blinded. So this trial actually started in 2018 and was going on, and COVID happened. So after we got rid of some bad scan data, we had actually really high adherence. Our initial target was 236, powered for a genetic interaction. But we had clean full data on 122, 64 to sugar, 58 to control. So no differences in BMI, no changes in BMI, no differences in self-reported food intake at baseline or during, no differences in physical activity over the intervention at baseline. We had a couple other measures, no differences. Our randomization worked. So again, you've seen this one before, except obviously we have brands in there. And so we tested them specifically on the sugar sweetened beverage taste, not the unsweetened beverage taste, which is a big kind of debate between whether we should do the beverage that they were assigned to or the beverage that was the test beverage. We elected to do the latter. But they did also get like a separate cue for that tasteless solution, just to not confuse anything. So the analysis on this, some of our results. So this is just the passive effect of the intervention. So control versus active, baseline versus follow-up. So a 3-way actually, and this was a BMI interaction. So it's actually like a 3-4-way interaction. And what we found was just greater insulin putamen response to the brand, nothing to the taste. And that interaction, which you can see in B, is basically showing that higher BMI to the sugar sweetened beverage had a stronger effect, and lower BMI to the unsweetened beverage had a weaker effect in this insular and striatal response. So stop signal task, if you're not familiar, I know we talked about it a little bit um, before, but basically we made a brand specific one, and we performed this to the sugar sweetened beverage brand and the unsweetened beverage brand. Um, and so your goal here is to respond as quickly as fast as you can when you see the brand, but withhold a response if an X appears. And the better you're doing, you guys are way too young for the GRE, but the better you're doing, the more challenging it gets, like it's an adaptive task, right? So it makes you feel worse the better you're going, but that's, just personally. So in this case, we weren't necessarily looking at the stop signal reaction time or anything like that, because we wanted to mimic Schultz. And so we just really wanted to look at reaction time as kind of this implicit like approach behavior. It's not completely implicit, but implicit like approach behavior. So how fast are they responding? And what we found was sugar sweetened beverages did decrease reaction times. So increase kind of that implicit motivation. And that was, you can see on the left, clearly no differences at baseline. You see a shift in the distribution and also the number. They are, the two groups are significantly different at follow-up, and there was a significant decrease in sugar sweetened beverage time, like corrected Bonferroni, these kind of things. We wanted to make sure that wasn't just kind of a random artifact of just kind of impulsivity or lack of attention to the task. So we looked at the standard deviation, and clearly, like there's no difference there. So that was nice. We're pretty confident that finding was true. Even more so, pilot study that I did without a control group, we found a similar effect. So we replicated the effect, which is it's always good, especially if you're doing imaging or behavioral work. So the next thing that I wanted to look at was to see if this change in reaction time corresponded with brain response to the brain that we had in the passive fMRI paradigm. So we're trying to mimic this Schultz pattern. And so in this case, we used an ROI of the nucleus accumbens because that's where the Schultz work was done. And you can see the ROI is outlined in black, significant voxels using randomized permutation correction for multiple comparisons is the activity with the lighter colors being stronger activity. So we did see that faster reaction times are associated with nucleus accumbens response to the brand. And so that's really nice. It parallels some of the primate work, which is always comforting when your stuff lines up. The reversal learning paradigm that I mentioned, we only did this at follow-up, so we didn't compare it to a baseline measure, but that was kind of by design because we didn't want to skew responses due to practice. Not that they wouldn't necessarily remember 3.5 weeks later, but that's just the way this one landed. I think we're gonna do pre-post on the next study. So the goal here is to choose between the two brands that are presented simultaneously. And if you get it correct, you get the sugar sweetened beverage. And if you get it incorrect, you get the unsweetened beverage, right? So obviously, you go up there, they've been drinking theirs forever. They know it's sweet, they know it's not. So this shouldn't take any learning. They should know this at the intervention. And so they flip, that's kind of great, that's kind of great. But after 2 to 4 correct responses in a row, the sugar brand contingency switches. So you expect an error on the first one, a probabilistic error that's going to happen unless they're guessing. And then the question is, oh, they got another error. How quickly do they adapt, right? And so how quickly they adapt is kind of our main outcome. And it's under the metric of reward accuracy overall. And that we kind of consider this sugar reward seeking type behavior. And we actually found that there are differences between the sugar group and the unsweetened beverage group. Keeping in mind, this is follow-up only. So those with the sugar sweetened beverage showed higher reward accuracy. Yeah, so you can see kind of the means. This is, it has to be distributions based on varied numbers of which events, because the number of whichever events, whether it be sugar sweetened beverage or non-sugar sweetened beverage, depends on their accuracy somewhat. And you can't achieve 100% unless you're really, really lucky because of that probabilistic error. So as you'll probably see a pattern, we wanted to see if this reward seeking mapped onto the brain. And we didn't have enough events, considering that they switch and there's inconsistent numbers of events and the paradigm was relatively short, to use your typical kind of fMRI evoked response like we talked about, like the donuts versus the Brussels sprouts or whatever they were. As opposed to that, we used connectome predictive modeling to do this. And if you're not familiar with that, you're taking, even it is event related data, you're kind of aggregating across the full paradigm. And it's a machine learning approach. And the goal here is derive a functional brain network that predicts the behavioral variable. In this case, would be reward accuracy. And just to be a little more specific, some people could debate whether you should say predict there, because this is not an outside sample that we're trying to predict accuracy in, or predict the network in, but we'll just use predict for today. So what you end up doing is you take reward accuracy scores. You have regions of interest across the whole brain. I think we had 268. And you're looking at the time course of each of those ROIs or nodes. And then you do cross-validation. And what ends up coming out of that is connectivity across these. So the higher essentially correlations, a higher connectivity between those brain regions. And so what we found here is that brain regions, firstly, if you see the graph, the sugar sweetened beverage people actually were able to, that analysis was able to significantly predict a network. And in the unsweetened beverage group, it was not. So, and then you look at this on the brain level, and these kind of things are always messy. So I'll break them down. Probably easier to look at this here, darker colors, more connections. Primarily it's kind of cortical, but you can see some medial work. But just looking at this in terms of not necessarily nodes, but previously established canonical brain works, you can see the colors darker is more connectivity. And the last plot that you can look at this is this type of chord plot. And that also is really tough to digest as I'm flipping through. So let's break it down. The most connected, both within network and between network connectivity, was the subcortical network. So again, we're talking striatal, insular, and some various midbrain, these kind of things, or not midbrain per se, but various other subcortical type regions. And you can see highly connected across the board, and then also within network. The second was more sensory motor, and this is somatosensation, which is you look at these are like post-central gyrus if your brain region, and that's really common because it'll encode taste and mouthfeel. And then obviously like motor processes, because they're responding in there. So it would make sense that those are strong. And obviously they're correlated with hedonically motivated behavior as well. And lastly, the third one was the salience network. And typically this is interpreted as being associated with stimuli salience and task switching, and then also just kind of general attention. And the last thing that we wanted to look into on this, and I mentioned it first, actually describing the design, is looking at those explicit hedonic ratings. So how much do they actually like them? Like, how pleasant is this beverage? How much do you want to consume this beverage? And, the liking, wanting questions. on a scale of -100 to 100 and see if there is a change in liking, because we've seen changes in behavior, whether that be kind of this implicit approach behavior or kind of slightly more conscious reward seeking behavior. So does that relate to changes in preference? Like, I like this more. It doesn't. So explicit liking of the sugar sweetened beverage or the unsweetened, the certain sugar sweetened beverage was not impacted by consumption for liking or for wanting. So we did also analyze this for the unsweetened beverage, and there was a difference in the unsweetened beverage group that they increased their liking and wanting over consumption of that. And part of the reason we think that is an unsweetened kind of novel fruit flavored beverage is a lot less familiar than something is like, it's a different flavor, kind of like Kool-Aid. So if you look at food neophobia literature in kids and picky eaters, those picky eater kind of work, it takes roughly 8 to 12 exposures before they start developing a preference. So if you consider they had 21 exposures and it's kind of a novel food, you would expect that to creep up. At least it would make sense. No changes in the sugar sweetened beverage group there. Sweet. So just kind of wrapping up, I flew through stuff, so happy to talk on things. But I think one of the most impactful things that we've come up with this last study and some of the work in the back half of the talk is oh yeah, that's wrong. Okay, cool. Just pay attention to the top. We think we found kind of at least a mechanism of food branding and advertisements. And this consumption of sugar results in this brand sensitization, like a hedonic target, which is a term used in preclinical research. Let's skip that one. There we are. And how this occurred was we had that increased implicit approach behavior to the brand. And if you're into decision making, my colleagues and I are kind of going back because we did not directly assess goal-directed and habit-based decision making, but this increase in reaction time is somewhat reflective of habit-based decision making, creating a kind of a habit, and that habit-based decision making really reflexive, really quick, insensitive to changing reinforcement structures. And we did see that this, whether it be decision making or just implicit approach behavior, was encoded in the nucleus accumbens, replicating some primate models. And we also saw this kind of increased sugar reward seeking, which is completely the opposite of habit-based and tends to be a little more goal-directed, in that it's goal-directed can be more characterized by having a lot of cognitive flexibility, or you update to these changing reinforcement structures. So when you had that during that reversal learning parent time, when you had that switch, people that drank the sugar were more apt to be flexible to update their choices. And there's actually some really, if you're in decision making, there's some really interesting work by Todd Hare lately and a couple others that are questioning whether habit-based and goal-directed are dichotomies, or they actually kind of work. One of the theories was goal-directed, once it turned into a habit, kind of turned into habit-based. But a lot of the work, including, if you know, the two-step task, which tries to differentiate between the two, people actually are in flux between and use both, specifically if there's a cue involved. So our reward-seeking portion was predicted by a network that was integrated in subcortical, somatosensory, and salience regions. Again, and this is just in the sugar group. And lastly, this occurs independent of explicit changes in preference or desire. So there's a couple models, and it's becoming more and more accepted or more popular that a lot of the drive and a lot of the striatal response is actually driven by gut signaling of calories. you have taste receptor, you have your sweet taste receptors, which will signal your brain calories and possibly like reward if that. But you also actually have sweet receptors in your gut, and but there's also gut through the CNS up to the striatum, and it's hypothesized that that gut CNS push to the striatum is occurring at not a conscious level, whereas your sweet taste receptors is more preference and sweet taste receptors and kind of sensor oil flavor and smell. So yeah, and then the last question that, on this that I'm always kind of curious to people's thoughts, and oh, none of this is published, so I'm always happy to, so not peer reviewed, but would love insights on it, is one could just say that we experimentally induced a habit. These are all characteristics of habit, which isn't necessarily a bad thing. You take something that's rewarding, you do it a lot, you would expect these patterns to happen. So we commonly, like I presented data on sugar, and I framed it like excess consumption of sugar is really bad. We don't know how much is too much, but let's say you flipped it and you did it with, I don't know, some low-level exercise. And the person's novel to exercise, they really don't like it in the front, and maybe they start to enjoy it, and then they're more responsive to running shoes or a New Balance ad or something. I don't know. So one can make a case that this isn't that groundbreaker fantastic, but it's just like a natural process. Although the food addiction people will love it. So at that point, I'm just gonna wrap up and thank my lab, particularly those highlighted in purple, who've done the vast majority of this work, and our other colleagues. My lab's a bunch of goofballs and it's wonderful. And I also wanted to thank Jessica, Joe, and Brock for getting me out here. Really appreciate it. And we are hiring. And because I'm moving places, my website is down, but that's my email. And you can find me on Blue Sky if you want. Don't bother with X, I'm almost done. So yeah, happy to take questions. Yeah. We have not, although in this study we haven't looked. In that like initial trial, which was much smaller, we did, and it lacked a control group, a peer control. So I did look at that. There wasn't, but that was also a number of years ago, where different analysis software and sample size of like 40 or 50 or something. So that's something we're testing. There's a couple other things that we're testing, individual differences in one trial learning, or we have some genetics, but probably underpowered for that. Yeah, yeah. 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 So I think, and yeah. 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 Yeah, no, and that's why I mean, the first step in that when we kind of moved from studying, variable weight status versus behavior. And actually in the RCT, I was trying, I wanted to get individuals with a very tight low BMI to see if we could cause this effect. And reviewers really didn't like that. So we took that part out.
Worry about specific reasons?
But no, I think it depends on who you are.
And how did that change your graduate school experience? So I know Emily just kind of answered that, but anyone else have anything to add?
So yes,
Go ahead, Lauren.
sorry. My program is very research heavy. So I did have a pretty kind of more general idea of the kinds of things I wanted to study. I knew that I was interested in negative symptoms and psychosis. Affective and social were kind of my jam. And over the course of grad school, I was able to hone in more on specifically what my spin on that would be. So I started out
Christina, you're not muted.
How my advisor leaned. And then I started to get more social and then thinking about other fields and bringing in how other people think about social processes and then apply that to psychosis. So it really just like whittled into being more and more specific over time until I really landed on.
2 talks for 1. No, so it's totally fine. No big deal. No, I think especially with some of that stuff with the gut signaling, I think that's going to be a big differentiator. It honestly kind of depends on who you are, because there's a lot of camps that are saying, sugar elicits a greater response than cocaine rewards in mouse models. And maybe it does, maybe it doesn't. And those tend to be kind of the food addiction camp. But there's got to be there's got to be more going on. I think the brain part is similar, and I think that just kind of operates on maybe like a hedonically motivated behavior, liking, wanting level. But I think there are differences, and I think I do think it's driven by hormones, and which obviously sex differences would be there. It'd be really interesting on GLP-1 agonists, of course, because that's popular. But those kind of things. No, that's what I wanted. Yeah. Yeah, so we did control for that, except for that one three-way interaction on the front end. And there were no effects there. I did want kind of a leaner. I think the average BMI was like 27, like our high BMI was 32. So we were skewed very lean relative to the population at large, but not scientifically where I wanted to be. And honestly, that just kind of, their comment was good there, and I'm not like picking on the reviewers. I went to a more public health facing, and they're like, you need to make the results more generalized. And so it's a question of scientific rigor versus generalizability in that case, but yeah. And frequently, like, there's a conversation about food addiction. Even the people that are hardcore food addiction people are certainly backing off the term. But there's a great paper by Alex DeFelice Antonio lately that showed the evidence that we have as far as they're very pro food addiction, and the evidence at the time that the Surgeon General made cigarette smoking addictive, called that addictive, and they actually match up pretty well. That at that time we have the same amount of evidence, and at that time they called smoking addictive. Cool. Oh yeah. Yeah. And so that's where, and kind of one way to parse that out would be artificial sweeteners, if you can match it, or you do the 4 kind of parametrically, you do the 4, like sweet plus calories, no sweet calories, sweet no calories, no sweet no calories, right? Because then you're fooling the sweet taste receptors, but then there's a gut response signaling a lack of calories. So it's actually kind of a weird gut brain prediction error. Like, oh, here come calories. Oh no. Which some people think, they call it the mismatch hypothesis, which, some people think that that would increase consumption of foods or sugar sweetened beverages in particular. And I mean, and actually kind of paralleling that, one of like the gut brain or kind of physiological brain connections was postprandial. So after consumption of a sweet thing, you look at the rise in blood glucose, and the area under the curve there predicted NAC response to food cues. So it's like that kind of, those kind of associations. No, yeah. And so what we're trying to do, it's hard to figure that out and maintain adequately powered. And that just kind of becomes a grantsmanship game, because they'll ask for 4 cells, and let's say we even get 200, and then out of the 50 per cell, we'll probably lose 5, 10 people, and now you're at 400 per cell, and I get a little bit uncomfortable that low. Anything else? All right, well, thanks very much, team. Enjoyed it. Have a good one.